This is Laura Fulford welcoming you to this Bite-Sized Bio web seminar, which today is titled The Basics of Intracellular Cytokine Staining. Today's webinar is presented by Barry Moran. Barry is head of flow cytometry in the Trinity Biomedical Sciences Institute in Trinity College, Dublin, Ireland. He began to develop his expertise in the Scripps Research Institute and USCD in San Diego before returning to Ireland to take up his current position in 2005. He's a director of the Cytometry Society of Ireland and has strong links with the international flow community, and is currently undertaking a part-time PhD in immunology aimed at advancing cytometry techniques in the study of immune regulation. We will have a brief question and answer session after the presentation. Please type any questions that you have into the questions box, which appears on the right-hand side of your screen, and I'll put them to Barry at the end of the webinar. So now over to you, Barry, for the presentation.
Thanks very much, Laura, and I'd like to thank Bite-Sized Bio for the invitation today to give this webinar. So for the next half an hour or so, what I'd like to go through is firstly maybe to illustrate why I think intracellular cytokine staining is one of the most exciting techniques in flow cytometry, and maybe a little bit of where my particular interest is. Also, I'd like to go through a little bit of the breakdown of the technique and some of the various aspects that relate to it. Then also some of the complementary applications or complementary techniques that tend to be allied to cytokine staining. And then maybe towards the end, some tips and some hints that I've accrued that may help you along with some of your work. So, as Laura spoke about, I'm the manager for the flow cytometry facility here, and with that is involved with many different types of cytometry techniques. But with some of the work I do and with the principal groups I work with, we're interested in immune regulation and how it affects or how it relates to autoimmune disease, and actually in cancer and tumor immunology also. So if we think of a healthy individual and a healthy immune response, what happens is when we see a pathogen infection, our body and our immune system reacts to that, and once the infection is clear, our body goes back to homeostasis, where that inflammatory response is suppressed and back to a balanced thing. So in immune dysregulation, where this isn't controlled, what we see is in this type of seesaw expression here, there's a tip to where the pro-inflammatory response is uncontrolled by that suppressive side, and this leads to autoimmune disease, from where people maybe know it as psoriasis or arthritis, rheumatoid arthritis or multiple sclerosis. And you know, we're seeing that this type of immune diseases are really relating to more and more common disorders. Likewise, we see a similar but somewhat opposite in immune regulation as it relates to cancer. So normally in a tumor, our bodies mount an anti-tumor response, but unfortunately the tumor itself produces suppressive and regulatory molecule where it dampens down this pro-inflammatory or this anti-tumor response, and in that way we see this seesaw is essentially tip the balance the other way, where we unfortunately have this anti-tumor response is suppressed. So a lot of the work we do in our department is trying to address that that imbalance, and where we can look at improved therapeutics to this. So a lot of the cells that we study for this, and the output is by cytokines. So firstly, we need to think about multicolor flow cytometry, really to identify the cells involved. So if we think a lot of the immune cells involved, where our particular group are interested is principally down here in this T cell. So within all the immune cells involved, this is a small subset. So that's our T cells. And then if we think within our T cell population, again, which our group are particularly thinking, all these different T cell subsets, we're principally interested in these helper T cells and then these regulatory T cells. And then if we further look into that, we have all these subsets of these helper T cells and these regular T cells. Maybe some of you have heard of Th1 cells, Th17, Th2, follicular helper T cells, and then our natural and inducible Tregs. And all these subsets of cells, there are various surface markers that we can identify these cellular populations, but also we need to identify them by the cytokines they produce. And also, if we want to study their function, we need to look at the cytokines that these cells are producing. So let's think about cytokines firstly. So cytokines are just small proteins that control and orchestrate the immune response. They are secreted by the cells and they interact with nearby cells, and they encompass a lot of terminology such as interleukins, interferons, chemokines, and growth factors. They have various functions from downregulating or upregulating transcription factors, induction of other cytokines, suppression of other cytokines, and even feedback where they suppress their own effect. So, as I hope I've shown you in the first place, we're very interested in studying cytokines, not only to study the cells in particular, or to identify our cells of interest, but then also to study their function and their interaction with other cells. So some of the classic methods of studying and analyzing cytokines, and the first example is ELISA, which you may be familiar with, which is a way of studying the overall cytokines within a solution or within the media, and that looks at the overall population. Likewise, such things as Luminex bead arrays or cytometric bead arrays, which are essentially a bead-based ELISA, which is a cytometric technique, but we're essentially looking at soluble cytokines binding to beads and then seeing the signal from that. And then also what we can do is we can lyse a cell population and look at the RNA expression level. Again, we're looking here at the messenger RNA, so that's really the message level rather than the protein level. So that may not be satisfactory. But again, in all these three examples I've given you, this is looking at the overall population of cytokines within a cell population. So we're not actually seeing which particular cell is producing which particular cytokine. And that's a very important technique that we'd like to be able to do. So some of the single cell techniques that we have available to us now is a CSA, which is a cytokine secretion assay. And in this, the cytokine is released from the cell. We have capture antibodies on the surface of the cell. They essentially capture that cytokine as it's being released. And then we can actually enrich that cell and identify that max. That tends to be just one cytokine at a time. It can be a little bit technically difficult, and as far as I know too, there's just one company at the moment, Miltenyi, that do this technology. In my experience, it's not a very used technology, but it is something to be aware of, and I guess the greatest thing about that is you can keep the cells alive and somewhat untouched. There are also reporter proteins where we can transfect tagged cytokine proteins in transgenic mice. Obviously, this is just a facility within mice rather than humans, and there's obviously a lot of technical and somewhat expensive development costs to develop these mice. And then we get down to what we're talking about today, intracellular cytokine staining, and that's where we block the cytokines being released from the cells, and then we tag it, and we can tag multiple cytokines at once, and we can tie it in with other staining features so that we can look at a lot of things simultaneously. And again, that's an important technique that gives us a lot of advantages. So if we think about, again, just to reiterate, normally a cytokine is released once it's expressed within a cell, it's secreted from the cell where it interacts with nearby cells. But in intracellular cytokine staining, what we want to do is we want to block the release of that cell so that we can characterize each individual cell and the cytokines being produced from that cell. We can use this with most, or if not all, immune cells of use. In particular, it's used a lot with T cells, also monocytes, macrophages, dendritic cells, granulocytes. And I'd imagine most cells that are, as far as I know, most cells that are producing cytokines are amenable to intracellular cytokine staining. And then, as I spoke about, we can also tie this along with cell surface staining, so we can identify cell markers, and also maybe we can use it with cell activation markers and other features that we can use that are expressed on the cell surface. I guess two things to note that are different to your classical, maybe more basic cytometry staining. The first thing is, again, to reiterate, we have to block the release of that cell. Usually, if we're doing intracellular protein staining, the protein isn't naturally secreted out, but with cytokine staining, we need to block the release of that cell. And then in order for us to stain and tag intracellularly, we need to be able to permeabilize the cell membrane so that we can allow the access of that antibody into the cell to stain its specific cytokine. In normal case, if we have our cells of interest, whether they're ex vivo or in culture, if they're just resting cells, we usually don't have enough cytokine being produced or expressed to really be at a detectable level. So usually what we have to do is activate or stimulate or prime these cells to be able to detect the cytokines and be able to detect the response. The ex vivo, if we see down here in nature, we have our first signal and our second signal, and we can mimic that in the lab where we can use, maybe something like anti-CD3 and anti-CD28. We can use an antigen specific to detect an antigen specific response, but again, that's usually at the lower end of detection. So unless it's a very strong, potent antigen specific response, we may want to use some of these super antigens, whether they be anti-CD3 and anti-CD28, maybe some particular cytokines itself, which will stimulate the expression of other cytokines, or else we can use these super antigens such as Staphylococcus enterotoxin B or PMA ionomycin, or I'm sure there's various other examples, but it's something we need to think about. Maybe we need to activate and prime these cells to be able to detect a certain cytokine response. Now, how long this priming of the cells needs to be depends on a number of things. Firstly, it depends on the stimulation type, it depends on the cell type, and it depends on the cytokine of interest. So whether it's a very potent stimulation or a weaker stimulation, it could be anything from a 2-hour stimulation to maybe a 5-day stimulation. Different cells themselves, whether they be T cells or macrophages or dendritic cells, will need, depending on their stimulation type, will need different times. And then different cytokines need different time duration to express. Some cytokines are expressed very fast, other cytokines take days to express in sufficient numbers. So we need to really figure that out, and none of these are kind of absolute. They're all things that usually we need to optimize and test out in the lab and in vitro before we have a robust system. So I've spoke about blocking these cytokines. So normally in immune cell, the cytokine is released. In intracellular cytokine, it's imperative that we don't release the thing. We have to have this buildup of the cytokines within the cell so that we can detect it. There's two ways of, or two chemicals that we can use, one or the other, to block the release of these cytokines. First one is profeldin A, which is a reversible treatment, and this just redistributes proteins from the Golgi complex to the endoplasmic reticulum, and then monensin interacts with the Golgi transmembrane transport to inhibit the release, and this is an irreversible treatment. So really it's again, it's a case of the cell type, the treatment type, the cytokine of interest, depending on which cytokine we want to use. Now, some of the things to think about when we're thinking about protein blocking are the activation steps. Firstly, both treatments can affect the actual expression of our surface markers or our surface proteins, as well as affecting the cell shape, as well as affecting what we call the scatter characteristics. So, how the cells scatter light, like our forward scatter and our side scatter. So these are things we need to consider. They also, both treatments on protein blocking or cell activation will lead to a lot of extra cell death. And again, the longer the incubation time or the more potent the stimulation, generally the more cell death we'll get. So we need to think about, optimizing our setup depending on the cell type, the stimulation type, and our cytokines of interest. Now, I just took this from the BD website of proposed protein blocking for different species and different cytokines. I would say that none of this, again, as a rule. I found in some of these cases for TNF in humans, the Profeldin A works perfectly fine. So I'm sure all of these things are really best optimized in your cell type and your stimulation type with ultimately in your hands. So, but these are things to be considered. As I spoke about this protein blocking, as well as cell activation, as well as all these other steps, can really lead to a lot of cell death. And this cell death can contribute to a lot of non-specific binding. By their nature, dead cells will take up antibody non-specifically and bind. And unless we have a control for this, we can get a lot of false positives. And in particular with intracellular cytokine staining, this can be a big issue. So it's really important that we try to exclude our dead cells from our analysis. Now, traditionally, the viability markers such as propydium iodide, which are DNA binding dyes, which we use in a lot of live cell assays, we can't use them in intracellular cytokine staining because by its very nature in intracellular cytokine staining, we need to kill all the cells or permeabilize all the cells. So if we were to use propydium iodide, they would essentially all stain positive. So this isn't an assay we can use. But fortunately, over the last few years, they've come out with these new amine dyes or fixable dyes, and these bind to the amine groups on proteins. Essentially, if we look at here in a live cell, you'll get a small bit of binding to the cell surface, and in a live cell, the cell itself will actually pump out these dyes, so they won't be able to bind intracellularly. Whilst in a dead cell, the dye can get in and bind a lot brighter, and you'll see that that's maybe a 10 to 50-fold brighter signal. So that's really a great tool that's benefited us in the last few years. There's a number of commercial dyes out there. I've just listed a few. I think Zombie is from BioLegend, Live Dead is from Life Technologies, Ghost is from Tombow, Fixfire eBioscience, FESBD. But again, I'm sure there's a lot of other examples that are commercially available. But this is something we always need to consider about tagging for viability to exclude our dead cells from our analysis. So that's really just our stimulating and identifying our dead cells. But let's think about our other staining protocol. So we block the release of the cells, but now we want to do two steps. The first thing is we want to fix the cells, or what we call fix the cells, which is really to fix the intracellular proteins in place and to conserve antigenicity, so their antigens are presentable to the antibodies. And then we also want to permeabilize the cell membrane so that in a normal healthy cell, the cell membrane is impermeable to these conjugated antibodies or these antibodies. So we need to permeabilize that membrane so that we can get the antibodies entering through the membrane. So paraformaldehyde is essentially the chief or the principal, or probably the most, not only used fixative. An example of a permeabilization or permeabilizing buffer would be saponin. Others would be Triton X, Tween 20. There are some people prefer to make up their own in-house, but there are many commercially available reagents from a host of different companies. So these are readily optimized. So if you can afford it, I would suggest that the commercially available kits are the way to go. So let's maybe take a look and maybe summarize what's involved in, or perhaps review what's involved in intracellular cytokine staining. So if we take our cells ex vivo or after culture and we have them just resting cells, we might have a small bit of cytokine being expressed within the cells. What we'll do is we'll stimulate those cells, and then we'll also protein block. Now, it's important to mention also that it depends really on the system in place. Sometimes the cells work best at just being blocked for the last few hours. Sometimes it works fine for the stimulation and the protein blocking to be treated together. And again, this is a kind of an optimizing thing. In my hands, for a lot of applications, stimulating as well as the protein blocking at the same time is fine, but you might find that protein blocking leads to excess cell death. Again, things to consider. But after we stimulate and keep the cytokines in place, we'll find that the cytokine expressing cells, once stimulated, will be expressing a lot more cytokines within, and again, we block that so there's no release into the extracellular milieu. The cells that are resting, that aren't primed in the first place or aren't expressing the basal level of cytokines, at this point will still not express cytokines. So it's only really the cytokine expressing cells that we increase the signal from. From here, we use a viability stain to exclude our dead cells. And at this point, it's a good place for us to also, before we fix or permeabilize the cells, to do the surface staining, because this will minimize non-specific binding. So any of our surface stains that we require at this point we'll add, so we can see our whatever antibodies we're using will bind at this point. Here then we'll fix the cells, so we again preserve the antigenicity. We'll permeabilize the cells then, where we permeabilize the cell membrane to enable the antibodies, once staining, the intracellular staining, to access the cells and bind brightly. And then at this point we'll actually analyze the cells on the cytometer. Obviously there's all wash steps at every step and various other situations, but this is just maybe a nice summary of what's involved in the key steps. So I just have an example of intracellular cytokine staining that I did a number of weeks ago. Now this is somewhat complex. Intracellular cytokine staining could be perhaps maybe 1, 2, 3 color to 15, 16, 17 color assay. In this case, it's a 15 color assay that we performed on our LSR for TESA. We have a number of surface markers, number of intracellular markers, and then I've just listed their conjugated fluorochromes to it. And if you are doing a panel, maybe working out and having a plan like this will help. You can see the setup of the cytometer and then on which detector each fluorochrome is and what the antibody for that fluorochrome is. And then to give you an example of some of this, the post-acquisition or the analysis gating that I've done, there's no right way or essentially wrong way of doing this analysis. It's really up to the individual, and once you really follow a particular logic of gating, there isn't necessarily a wrong way of doing it. But in this case, this was from a tissue with a lot of firstly a lot of dead cells and also a lot of non-immune cells or non-leukocytes. So because I was interested in maybe zooming in on the immune cells, firstly the leukocytes, I've looked at a CD45 pan-leukocyte marker versus my viability marker, and then I pulled out the population that are positive for CD45 but negative for my for dead cells. So my first step is to pull out my viable leukocytes. Then I've put on my classic forward versus side scatter to identify my lymphocyte gate. I've done that, and then I've drilled down into that population, and I've excluded the few doublets where two cells have stuck together. I've excluded that, so I've had what we call a single cell gate. And then from my single live lymphocytes, I've looked at CD3, which is a T cell marker, and CD8, which is a T cell subset marker, and I've identified my cells that are CD3 positive, CD8 negative, and then also CD8 positive, CD3 positive. And then of these cells that are CD3 positive, CD8 negative, I've drilled down into this population, and I've identified on another marker, which is TCR gamma delta versus, again, my CD3. I have a population here that are CD3 positive, gamma delta negative, and I have a population here that are gamma delta positive, CD3 positive. So ultimately in here, I've kind of zoomed down, and really this is almost my starting point where I can identify three subsets that I'm interested in. Firstly, these are my CD8 T cells. These are my gamma delta T cells. And then somewhat by default, these are going to be my CD4s because these are CD8 negative, CD3 positive, and gamma delta negative. In this assay, I wasn't able to use a CD4 marker, essentially because the CD4 antigen is downregulated in this assay based on the stimulation. So again, it's really me identifying the CD4 by knowing what it isn't, that it isn't CD8, that it is CD3 positive, but it's CD8 negative and gamma delta negative. So let's just in this case, let's just zoom in on our CD4s. So, and this is almost our starting point to look at our cytokines. So, we can look here, and in this case, I've, the 8 cytokines, GM-CSF, IL-17, TNF, and I can see the output from there. And if I want, I can zoom in further and zoom in further. But maybe that's just a good example to show the first few. So that's intracellular cytokine staining in a nutshell, but usually we use complementary or related techniques alongside that. And one thing is, and maybe it's an interchangeable term, but is intranuclear staining. So rather than cytokines, often we're interested in various transcription factors, and certainly in immunology, it's often, one of the key ones would be FOXP3, which would be your regulatory T cell marker, your classical transcription factor for regulatory T cells. And to enable the antibodies enter the nuclear cassette, we need to, or the nuclear membrane, we usually need a stronger permeabilizing buffer. The saponin in particular isn't strong enough, so we need to use something more potent. So we can use, again, we can prepare a particular buffer, but again, there's usually there's commercially available ones that we can use. So there are either general ones, or there's often antibody specific. So there could be like a FOXP3 specific intranuclear staining buffer. Usually whatever is fine for FOXP3 is fine for any other nuclear protein, as far as I've found. And I've also found that you can use these intranuclear fixation and permeabilization buffers with regular intracellular cytokine buffers. So you can use these particular buffers for intranuclear and intracellular cytokine staining all at once, and surface staining. So that's a nice technique that we can add to it. Another example that I thought I'd bring up was looking at detecting phosphoproteins, which is a relatively new technique. So protein phosphorylation is involved in apoptosis, cell signaling, stress responses, various cytokine pathways. And classically, there was a lot of slow and even potentially dangerous, like radiometric assays or blots. But now we can use these phospho-specific antibodies that are commercially available through a number of different companies, and a lot of different phosphoproteins are commercially available now. They tend not to be very bright, so you don't see a nice 50, 100, 500-fold increase in fluorescence. It's often 1.5, 2, 3-fold difference. And often you see this data presented as overlays rather than in your classical dot plot. So it's you don't see as nice a fold change or as nice a signal as you might like, but it is a very kind of well used and well characterized technique now too. And again, we can use this alongside cytokine staining, and it's just, I guess, this is one thing, I guess, with all of them, that we really need to think of the experimental controls and perhaps isotype or various treatment controls that we might want to use. Because we don't see a large effect, whether positive or negative, we need to consider that. Okay. Another new that you may not be aware of, a new technique, which is what we call RNA flow, or detection of RNA transcripts within a cell, which is really done by FISH, just in situ hybridization, where the cells are fixed. Then we have a gene-specific label. That label then is amplified, and then we can use fluorescent label probes for that. I haven't used that myself yet. I've heard that it can be technically quite complex, and it's, at one stage I heard too, it's like a 16-hour protocol. So if it's, I don't think it's something that you can very easily work in, but it's certainly a nice new powerful technique. At the moment, as far as I know, it's just eBioscience that have this technology, and it's called prime flow RNA. But again, it's something that you should be aware of, or that you may be aware of. And then I've just put in this also, you're probably used to the various analysis softwares, or how we interpret the data post-acquisition. But then I think, and in particular with intracellular cytokine staining, SPICE software is a nice way of presenting this data. And we're looking at possibly the differential expression of a lot of different cytokines within one cell. It can be hard to present that data in traditional flow presentations of flow plots. So Mario Roeder's group in NIH come up with this SPICE software that's freely available, and it's just really it's a way of interpreting data which has to be analyzed via FlowJo software, and it's just a way of presenting it really on a plot where we can present whether this is our total cell population. So we can see here that we maybe nearly 25% are only expressing one cytokine, about the same or two, and then 3, 4, 5, and then of let's say in this case where they're expressing 4 cytokines, we can see what of those 4 cytokines they are, and we can relate that to this outer arc legend. So it's just a nice way of presenting quite complex data, usually cytokine data, on one plot, and that's maybe somewhat accessible. So some of the considerations that we need to think about, maybe that's worth mentioning in cytokine staining. Firstly, and maybe it doesn't need to be said, but it's not suitable for traditional sorting. That's assuming that you want to sort. Usually we do cell sorting because we want to work with live cells. We want to do downstream live cell applications. The technique of intracellular cytokine staining by default kills the cells because we're fixing and permeabilizing the cells, so they're no longer viable. As I mentioned before, we need to think about the activation type and the incubation time. There's no automatic answer for this. It really is what works best in your hands, with your cell type, with your cytokines of interest, with your stimulation type. So you need to consider that. You need to also think about the sample preparation. For example, if you're working with tissue, in order to digest that tissue into single cell preparation, that may actually affect cell surface markers, and I've certainly seen that with various markers. Also, the activation can cause the downregulation of surface markers, and a classic one of that would be CD4 expression in human T cells, where if you stimulate them with PMA and ionomycin, it's downregulated, so it's no longer expressed on the cell surface, or certainly expressed at a much lower level and maybe at a non-detectable level. The fixation technique too can affect various fluorochromes, in particular conjugated fluorochromes, tandem dyes. So that's always worth considering. And then also you need to make sure that the buffers and the actual antibodies themselves are compatible. For example, some fixation or permeabilization kits are methanol based, and methanol can degrade certain fluorochromes, so you may need to be aware of that. As I mentioned before, we always should surface stain before fixing and permeabilizing, and that's really to minimize or to limit the amount of non-specific binding. So we're really only allowing that surface antibody to bind to its surface because we haven't permeabilized the cell. So again, maybe to overview, our best practice is we'll stimulate the cells, then and protein block treat the cells. Then we'll use a viability stain to identify the dead cells. Then we'll surface stain. Then we'll fix the cells, permeabilize, and perhaps as we permeabilize, we'll use our intracellular staining. Then we'll analyze following that. A couple of lines about actually setting up on your cytometer. Again, whether you're using 2 colors or 15 colors, some kind of quick tips to consider, or how I would suggest anyway, is firstly, when you're setting up the cytometer, run your single controls first. So your single cell controls, which are often just your single stained compensation beads, and adjust the voltage for each of these parameters so that you get a nice positive peak in the primary detector, and it's 2-fold brighter than any other. So for example, if we start with our FITC stained cells, that we can see a nice positive peak being detected in the FITC detector, or the 530 detector, and that it's brightest in that. And if it is detected in any other channel, that it's a lot less. Then we run all our other single cell beads and do the very same. And then at that stage, I would run my actual positive or my maximally stained, so maybe my stimulated or overactivated sample, where I know I'm going to get maximal expression of my cytokines and surface markers, and I want to make sure that that's all on scale. And then at this point, I'll acquire and run all my compensation controls, possibly my fluorescence -1 controls and what other controls I have, and then I'll acquire all my samples. So again, in a quick minute or two, that's really the setup. It may not be that easy in practice, but that's the basics. Some of the controls you might want to consider, and again, there's no comprehensive answer to this, but you may want an unstained treated control. Now the reason I say treated is because the activation or the stimulation may affect the surface markers, it may affect the scatter properties of our cells. So we really want to see just unstained. So everything, if it affects the autofluorescence in the cell, that we can control for that. Our single color compensation controls, I hope you're all clear at why they're necessary to adjust for spillover. Fluorescence -1 controls are an important control that we use. You may find that you want to use fluorescence -1 controls for all your fluorochromes, but in particular you may want to use them for your dim cytokines. You may find that an unstimulated control is worthwhile, and that's really where you should see basal level of cytokine expression. So maybe at that stage, if you do see positive expression, it may pose questions. And then also you want an experimental positive control, because maybe with your treatments you might find that you're not getting any cytokine expression, but you may not know if that's because of your treatment or because of the actual that's just the way it is. Maybe something went wrong with the experiment, so you should always have a positive control in your thing. Again, a lot of these are common to all cytometry experiments rather than just intracellular cytokine staining. And then maybe some other things you might want to consider is where you don't simulate with your Golgi block, so you don't have the protein blocker. So in that case, again, you should have basal, if not no levels of cytokine being detected. You may also want to use an isotype controls just to control for non-specific binding. Also, you may want to use non or unconjugated protein, which will compete for binding sites as a control too. And I'm sure you can add more controls to there. Just a couple of words about our antibody or our fluorochrome selection. So one thing, or the first thing I'd mention is you should always titrate the antibodies irrespective of what's suggested on the data sheet. It's not always the case. Again, it's what works in your hands, and what you'll often find is that the suggested concentration is way in excess. So firstly, you can keep your costs down, but you can also keep your non-specific binding down if you titrate the antibody. Good practice is, if you're designing your panel, select bright fluorochromes on low expressed antigens, and select the dimmer fluorochromes for the higher expressed, kind of surface markers or your kind of classic cell markers. If you don't have a lot of colors and you can kind of minimize the overlap, then try to select fluorochromes that are spectrally far away so that you minimize that spillover. And then if you do have to select fluorochromes that are quite close by spectrally, what's good to do is select them on markers that aren't going to be co-expressed on a cell. And I have, in this example, CD3 and CD19. So essentially CD3 is exclusively on T cells, CD19 is exclusively on B cells. There shouldn't really be cells that are co-expressing both. So that's something that we can use there. And then, you know, for your own experiments, it might be worth setting up this kind of panel. This is something that I've done out that works for my system, where I've kind of, you know, figured out what I think is fluorochromes that tend to be very bright, not so bright, then moderately bright, and dim. And then I've also worked out a lot of the common proteins that I use that might be highly expressed, moderately expressed, or low expressed. Now, some of this is trial and error that I've done myself. Some of this is, you know, that I've learned from colleagues, and then some of this is actually available. And I know BD in particular are doing a lot of work at the moment, identifying firstly these kind of stain indexes on the fluorochrome side, but also looking at the expression of various markers on, or various proteins on various cell types and kind of quantifying that. So, you know, if that's something that you think you can avail of, by all means. But it's, again, it's certainly when you're at your panel design, these things are worth considering. So hopefully I've given you a quick snapshot of some of the considerations and what's involved in intracellular cytokine staining, and hopefully illustrated that it's, once used well, it's a really powerful tool and a powerful technique for immunologists and cytometrists. If you like some further resources or further reading, obviously there are great resources on Bite Size Bio, not just on cytokine staining and other cytometry setups, but also, you know, various other biology considerations and interests and aspects. The next thing, this is my own website that you're more than welcome to access. The kind of the main tome that should be read, you know, are certainly the main reference book is Practical Flow Cytometry by Howard Shapiro, which is really the kind of world authority on all things cytometry. There is a free PDF at the Beckman-Coulter website. I find all the reagent websites or instrumentation vendor websites have really excellent resources, you know, particular to their own reagents, but also kind of general reading. There are spectral viewers for designing, you know, your fluorochromes, as well as panel designers where you're designing not just your fluorochrome but also your marker. There are various ones, again, associated with the vendors or else just with particular companies, or sorry, particular websites like Chromosite or Flourish. And then there are various, you know, just more basic training tutorials, not just in cytokine staining but also basic flow tutorials such as Life Technologies and BD. And at that point, I will finish up with the webinar, and thank you very much for your attention, and maybe Laura may have some questions for me.
Thanks, Barry. That was a really excellent presentation. We do have a few questions. If anyone else has a question, please feel free to post it in the questions box that appears on the right-hand side of your screen, and I'll put it to Barry. So the first question, Barry, is, does it matter what you stimulate the cells in?
Well, often, or traditionally, you know, people just use their kind of standard culture media, you know, along with whatever, you know, the super antigens they work. But there was a paper actually out, and actually I have a slide here, where traditionally what's been used is RPMI, which is your kind of standard culture media that's also topped up with your pen strep glute and your maybe fetal calf serum. But what's been seen now recently is that actually to get really optimal cytokine expression, you really need to up the calcium in there, and that essentially in RPMI there's been a deficit of calcium. So there is a paper that the actual first author, I think it's Jakobsen, but by the Thiel group, I think, in Germany, but it somewhat escapes me at the moment. But instead, they've either subsidized or added calcium into their RPMI, or they've also used this IMDM Iscove's media, which has extra calcium in it. And in that case, you see much more detectable levels of cytokine. And I actually did this a while ago. I kind of compared this RPMI, our complete RPMI media versus complete IMDM media. And, I saw this with a number of cytokines. I certainly didn't see, some of the cytokines seem to be comparable, whether it was RPMI or IMDM. But like in this example where we're looking at tumor necrosis factor, you can see it's almost a twofold, where we're detecting, this is just stimulated with PMA and ionomycin, but it's over 52% in IMDM versus our PMI. So that, I think that's really important, that people need to consider the media that they're using, and I would certainly suggest going towards IMDM media.
Okay, and do you stimulate and block the cells at the same time, or does it matter?
Again, it's in my hands. I often do. A lot of that is just time constraints and practicalities. Again, with blocking, often you'll see an increase in cell death. So you may want to consider just blocking for the last few hours. In particular, if you have a long stimulation or a long incubation period. For example, if it was a 3-day incubation, you mightn't want to block because it will affect the viability of the cells. So in that case, I would maybe just go towards the last, maybe 6 hours to block. Often for the blocking, 6 hours is appropriate. So, if you have a 20-hour stimulation, you can block for the last 6 hours. But to be honest, in some of my protocols, I have a 20-hour stimulation and I add the blocking at the same time. I add the profeldin, and I don't see a massive increase in cell death. So again, in my hands, but it's worth really optimizing, I think, yourself.
Okay, thank you. Another question is, can ICS be used in the case of developing T cells, as in double negative staining of T cell differentiation?
Sorry, say that one more time.
Can ICS be used in the case of developing T cells, as in double negative stage, of T cell differentiation?
Well, again, you're not going to use it for developing the cells. ICS is really just a method of identifying the cytokines being produced in a particular cell. So if you're trying to culture, I guess maybe CD4 negative, CD8 negative, that's really going to be particular to the cytokines, or the stimulation or the culture techniques you opt for. So really, cytokine staining isn't going to be a tool for your stimulation. It's really just a tool for identifying on the other end. You could certainly use, if you're interested in the cytokines that are being produced by those double negative cells, you can certainly use cytokine staining. Your surface markers may be, for example, CD4 and CD8, and you'll look for the cells that don't express either of those. So you look at the double negative expressors and then see what cytokines are being released for that. You may also, if you're looking at double negatives of CD4, CD8, they may be other cells that are of interest. So they may be, for example, gamma delta cells. They may be NKT cells. So there may be other markers that you can use for that, or certainly, again, you can just use the CD4 and CD8 and identify that. But it's not a tool to develop these cells. It's really only a tool to identify these cells at the end and to study these cells at the end. So I wouldn't really be able to comment much more on developing them or culturing them in the first place.
Okay. Another question is mononesin block possible for live animals before necropsy for tissue?
I think I've heard something about that, but again, with I think the problem with live animal treatment of that is, you really have to think about what else you're affecting. So you're not just preventing cytokine blocking, but you're preventing really the release of all proteins. So in a live animal, you don't know what other systems you're affecting. So it would be hard for me to comment because obviously you've thousands of proteins being expressed in that. So it may be a tricky thing to optimize. I'm probably just not best placed to comment.
Okay, and then one final question I think before we tie it up today is it possible to use freezer cells, or is it better to use fresh cells?
Yeah, it's possible. Once frozen cells have been, frozen in, appropriately, whether glycerol or DMSO, it's perfectly fine to, thaw them out and stimulate them. Maybe obviously, fresh cells are going to be more responsive. You're also going to get a lot less dead cells, but it's ultimately a robust technique now, and certainly I use it frequently, where I just thaw out the cells, kind of fat, just almost flash thaw them at 37 degrees, and then stimulate them. The only thing you'll really see is you might see somewhat less activation rather than your fresh cells, and again, much more dead cells. But it's certainly a robust technique.
Okay, we've had another question pop in, so I'll just, we've got time to answer this one. So
Sure.
this viewer says, I'm actually working with human gamma delta T cells. Usually without culturing, we have smaller amounts of cells. Do we need more cells for ICS than for usual FACS surface staining due to cell death? And what is the appropriate number of cells in a sample?
Yeah, well, it's some ways a kind of how long is a piece of string. The more cells you start with, the more robust your statistics. But if you can make, I guess I would say as many as possible. But I've certainly had tissue samples, and again working with studying gamma delta cells, where they're a lot less expressed maybe than CD4s or CD8s. And I may just have a few hundred cells to work with. You will see increased death in intracellular cytokine staining. Even, again, once they're fixed and perm, they're all dead. But certainly once you activate them and you block them, you will see increased death rather than kind of regular surface staining. So if possible, you can up the amount of cells to starting, but it sounds like, that's not a possibility. You may want to minimize, perhaps the controls, or, minimize the number of samples, so you kind of limit the samples, and then you have more cells per sample. But it's really a case if you can make do, with just a few hundred cells, and if you can get robust statistics from that. So all I would really say is maybe plan your experiment that you're not, you don't have unnecessary samples, so that you limit your sample numbers, and then you use as many cells as possible for kind of as few samples as possible.
Okay, thank you very much, Barry. So that's all we've got time for today. So thanks again, Barry, for a very illuminating presentation and a wonderful discussion. And finally, thanks to the audience for taking the time to attend and listen in. If you've enjoyed the webinar and would like to view the video recording of the session, please visit the webinars page on bitesizebio.com, where it should be available within the next 24 hours. There you can also see other webinars we have lined up for you in Bitesize Bio Webinars Festival. So until next time, good luck in your research, and goodbye from all of us at Bitesize Bio.